Okay. Thank you, Roy. And I'd like to start off by thanking you and Cheryl and Aaron and John for bringing us all together. I don't know if the audience appreciates what a unique opportunity this is to bring people together from academia, from industry, from government regulatory agencies and research agencies. There are people in the audience from advocacy organizations and payers. These are the people that we all talk about, and we imagine what they would say. We would imagine what their concerns would be in these situations. But today we have the opportunity to actually hear from them. So thank you all for putting this together. So what I'd like to do, and it's interesting to see the commonality of the themes that are emerging here. We're not looking at this as blind men trying to describe an elephant. We're actually all feeling the same thing and feeling the same parts and recognizing that this all has to fit together into something that's more than just the sum of its parts. So I think now the challenge is going to be for us today to define those questions that are most important for IOM and the National Cancer Policy Forum to really focus on and really dive in deep with all these multiple perspectives to try and identify what the actionable issues might be to move the field forward. And for those of us who imagine the obstacles, and we've heard about regulatory obstacles, I just want you to think about what you've heard from Liz and from Rick here. These are not people who are simply rubber stamp government officials saying yes and no. These are people who are, I think, among the most innovative that you'll find at the FDA or any regulatory agency in terms of interpreting the regulations in a way that's relevant to the technology that's emerging. And when I talk to my colleagues at Pfizer about their interaction with their counterparts to Rick and other parts of the agency, I want to point out how lucky we are to have somebody like Rick who's able to really take his experience outside of the federal government and apply that to the informed decisions that he's made and his group has made regarding appropriately evaluating new targeted therapies. So with that, actually, my approach is going to be a little bit different, and that is I'm going to go right to the questions because, frankly, I have questions, and it's really up to all of you to help me answer them. So these are the four questions. How can we integrate genomics and other platform diagnostics into drug development strategies that will meet regulatory requirements for companion diagnostics? Secondly, can the results of minimally invasive diagnostic tests be used to expedite drug development and inform therapeutic decisions? Third, how can we make the greatest use of N of 1 or bucket studies? And lastly, is it possible to define a preferred sequence or hierarchy of therapies for a specific disease? So what I'd like to do is actually address each one of these in order. So first, how can we integrate genomics? So I can tell you from our experience at Pfizer, we've recognized that the era of one-off diagnostic tests has come and is rapidly leaving us. And the reason is several. Number one, it's very cumbersome. You have to now select one platform. It may be a platform that is very good for diagnosing that particular situation, as FISH break-apart assays are for translocations like EML4-ALK. But what you then have to recognize is the fact that not every medical center around the world can do it very easily. And so now you've created a barrier for the diagnosis of these patients and a barrier for them accessing these therapies. The other issue here that any pathologist will immediately tell you is the limitation of tissue, especially in diseases like lung cancer where we're now seeing 10 or more actionable mutations. If you have one-off diagnostics, you're going to run out of tissue before you run through all those potential mutations, actual mutations. So where we recognize this change coming and we're saying, we have to multiplex this, and let's get 10 diagnostic platforms for lung cancer. Before we took too many steps in that direction, we saw that the era of genomics was coming along and omics of other platforms as well. And we said, before we start doing this, we don't want to set something up and then it'll become obsolete because this is moving so rapidly. And now we're engaged with other pharmaceutical companies and interacting with Liz and Rick's divisions at the FDA to try and figure out how we can rationally incorporate omics platforms in the use of identifying particular analytes from those for use in being the companion diagnostic. So again, it really is a reflection of the creative thinking that's going on in all parts. And so, that's the third question. How can we not only qualify a platform, but then be able to identify one particular analyte of hundreds to be able to demonstrate it has the performance characteristics that are absolutely essential in order to be able to reliably identify the patients who have that abnormality and exclude those who don't have that abnormality. And this is really a dialogue that involves all of our constituencies. Secondly, minimally invasive diagnostic tests. One of the biggest challenges that people in translational research face is the inability to get serial biopsies, to be able to, over time, evaluate how the cancer has transformed and changed from the time you first got the biopsy and saw that wonderful response to the time you seeing that same tumor just taking off and growing with that same therapy, sometime after a prolonged period of time, and other times after a frustratingly short period of time. Patients have had remarkable responses that last only for a matter of a few months or weeks. What's happened? Has that been a sampling error? Is it due to tumor heterogeneity? Is it due to something else that we can be able to detect earlier and then be able to take actions upon? And I think what's happened now is the ability through circulating tumor cells or circulating DNA for us involved in solid tumors, unlike the luxury of people who work in liquid tumors like Fred has all his life, to be able to actually assess that over time and to use that information to inform therapeutic decisions. To be able to see the emergence of a T790M mutation in EGFR mutant patients, and then even in advance of clinical progression, then the question is, if you intervene at that time, can you actually have a more effective intervention? And if you intervene at that time with something that hits that resistant clone, does it also mean that the original clone is no longer operational, or do you also have to continue to hit the other non-T790M clones? And I think the answer is you do, because if you have a drug that's just specific for that, then the tumor then grows, and actually it's the original clone that grows out. And then being able to look at this over time and be able to relate that. Is that actionable, and how would we evaluate that? What would be your endpoint that would determine whether that clinical trial of intervening early would be a success? How would you set your threshold? What would be your endpoint? Would it be measurement or eradication of those circulating tumor cells in that particular clone? Would it be the time until the tumor progressed? How do you measure progression? Is it clinical progression or appearance of a new resistant clone in the bloodstream? So I think a lot of provocative questions there as well. Third, how can we make greater use of N of 1 or bucket studies? So what threshold of evidence should be required to qualify a biomarker to be used to select patients for N of 1 studies? That seems like a pretty simple question, but very difficult to answer, because as you saw from Roselle, relatively few tumors have just a single mutation that you could focus on and say, that's the one, this is the appropriate therapy. But they have a multiplicity of abnormalities. And how do you place these into some sort of functional hierarchy? How can you know which one might be driving the tumor? Because you might have a very effective therapy, but in that particular genomic milieu, you might fail to see a response, whereas you could take a second patient who had the same abnormality, but in a different milieu, and see a response. So you actually might, if you don't see the totality, and Spiro was very eloquent in describing the kind of network that's being used at TGen and others, as was Andrea, being able to take that and being able to ask ourselves, what does that totality of data tell us? Now that's a real challenge for biocomputing and bioinformatics technology. Are we smart enough to put this into a biological hierarchy that's going to have clinical significance? Many of the bucket studies now, the master protocols, the MATCH trial of NCI, the Friends of Cancer Research trial in squamous cell lung cancer, the CRUK study in lung cancer as well, and the WIN consortium, just to name a few that we're aware of, are all really focusing initially on a single genetic abnormality. And so when is that really the best first step, or are we setting ourselves up to fail? And if we do fail, I think we'll have more failures than successes, but if we do fail, how are we going to learn from that? How are we going to be able to make this a learning process and maybe establish neural networks to be able to take all the failed trials of single agents and then help to use that to inform us in the next generation of combination studies? And then how do we develop a systematic approach? This is a daunting task for any group. I don't know if any single medical center, no matter how large, is able to undertake this alone. There's going to have to be a new paradigm established to be able to bring together cancer centers and networks here and internationally, using same platforms, same bioinformatic approaches, to be able to have a wide enough net to be able to identify sufficient numbers of patients to allow us to identify these patients in subgroups in a timely manner, test these therapies, and get answers. Because if we try to do it in a one-off or a small limited basis, we're going to take so long that the questions that we have posed are actually going to become obsolete by the time we answer those. So that's another pitfall that we have to avoid. And the last question is something more clinical, but I think it's really something we have to think about. Everyone is trying to figure out how can we identify signals of activity early and speed the development and approval of these drugs to get them to patients who need them as quickly as possible. The accelerated approval program that's been in place for 20 years at the FDA is a good example of doing this on surrogate endpoints, sometimes progression-free survival, sometimes even response rates. But as we have more drugs that are available, some drugs are in the same therapeutic category, have similar mechanisms of action, we have to be very careful about continuing to rely on these in the same way that we did 15 or 20 years ago, because the utility of these endpoints may change in different situations. So let's take response rate, for instance. I think everyone working in immunotherapy rapidly identified the fact that they actually got, in some cases, tumor growth before tumor regression. So the people involved in immuno-oncology got together and decided they were going to create their own response criteria, the immune-related response rates, which allows tumors to grow and continue therapy as long as the patient is well enough, and then to see the benefit later on. So we have to be careful about response rates in that regard. What about progression-free survival? In many diseases, a surrogate for overall survival associated with clinical benefit. We've been real champions of using progression-free survival. But think about this now. As you develop more analogs within a particular therapeutic area with a similar mechanism of action, whether it's VEGFR TKIs for renal cell carcinoma, or ALK inhibitors for ALK-positive non-small cell lung cancer, you may have to think a little bit beyond that, because if you have a very potent agent that can give you a very long progression-free survival with that one agent, but then when that tumor develops resistance to that agent, it confers cross-resistance for all the other agents. So you have a long PFS. So that means that it looks good compared to maybe the standard therapy. But then think about this. If you then take the standard therapy with a shorter PFS, but whose mechanisms of resistance are not as powerful and don't confer cross-resistance, you can get very meaningful second and third PFSs, which taken together give you a better outcome than with using the most powerful agent up front. This really is a challenge to the paradigm that we all grew up with, and that is use your most active agents first, don't save them. But now we're dealing with a different setting than one we grew up in, and where we have to look at this in totality. Could this change the way we develop drugs? Could this change the way we evaluate the benefit of new agents and decide where to put each agent? It might. It might also take a little bit longer to establish this kind of sequence, but I think it's going to give you a more accurate picture of what your drug does and how it fits into that treatment algorithm. So with that, I'll stop and open for any questions. Thank you.
Great. Thanks, Mace. Does anyone have any questions now? Yes, it'd be great if you could run to the mic. I'm sorry to push that, but they record the meetings and.
So we now have a large collection of experiments where large number of compounds have been tested in large number of cell lines, both by Cosmic and by the CCLE project and the follow-up to that by CTD Square. And I think one of the data that came out from that quite clearly is that there are very few, as you would expect in sort of monogenic diseases, association between activity of a drug in cell lines and actual underlying single genetic alterations. So if that doesn't work in cell line, imagine how much more complex it's going to be in patients. So I completely agree with your view of, maybe we need to think about complex multiplexed assays to determine that. So now the question is, it seems that, and I completely espouse your perspective. I think the thing is, how do we put together a group of people that really bring together the various perspective of academia, the public sector, and the private sector to actually work on this problem in a very targeted and dedicated way, as opposed to sort of just have a randomized filter that says, you publish, and at some point somebody will pick it up in industry, and this will be then potentially developing a drug. It may not work because they didn't really understand how the study was done in the first place. I think there has to be a better mechanism. I was wondering whether this is the right, appropriate time to think about putting together a little bit of a collaborative organism that can think about, because sometimes even as basic scientists, we don't even know whether the problems we're working on are actually translatable. And I think having your perspective would be incredibly valuable.
I think it all comes down to incentives, and I don't mean that in just the mercenary fashion. I really think about even academic incentives. At academic medical centers, you're often evaluated based not only on publications, but first author or last author publications for a number of grants you've had funded. And that happens if you lead an effort. And so there's a real, whether it's intended or unintended, a real incentive for groups to really split off and say, I can lead this effort. And this has advantages that others don't have for this, that, and the other reason. I think here's maybe a paradigm-breaking opportunity. And we all have to engage in that from all of our different perspectives to make sure that this works. Companies need to be more flexible regarding letting the drugs out of their control at an early point in development. Some companies won't provide drug for investigator-initiated studies until they're already approved. That has to change. We have to be able to recognize, and I think that Roy and Ellen Siegel and Jeff Allen and colleagues at Friends of Cancer Research are doing this very well through their initiative to have a master protocol or a bucket protocol for squamous cell lung cancer, and making sure that individuals are recognized for their contributions there. But I think once you can get over that hurdle, I think you've taken away a big obstacle to making progress.
One last question, Bill.
So you mentioned VEGF, which I think introduces another problem with respect to getting to the right combinations, which is, as you can well appreciate, the VEGF-R inhibitor, for example, that appears to be most active as a single agent, perhaps because it has additional off-target activities, may be precisely the drug that wouldn't lend itself to a combination. Conversely, the VEGF inhibitor that's specific enough to lend itself to a combination may never look very good in a head-to-head comparison with some of these first-generation inhibitors. So is there a workaround that we can get the right drugs into the combinations without always demanding that they perform well in a head-to-head single-agent comparison?
Yeah, I think it's a great question. You've increased the complexity now because you introduced multiple elements to the therapy. But I think that there are and there have to be. Whether there can be small clinical experiences rather than large head-to-head trials where you have enough patients to have statistical significance for each comparison, but rather by having small clinical trials and setting the bar very high, you can really establish that quickly.
Great. Thanks, Mace. Okay, our final speaker for this session prior to the discussion will be Dr. William Haidt, who's the Global Head of Research and Development at Janssen Pharmaceuticals. Prior to that, he was the founding director of the Cancer Institute of New Jersey, where he was Professor of Medicine. Bill.